Okay, to leave off where the last video cut me off, because I only have 15 minutes and I can never say all those things in 15 minutes, is that we have three genotypes. Let me get the pen out. So, homozygous dominant, which means you have two uppercase letters. Okay, so organisms that are homozygous dominant will show the dominant trait. Then we have heterozygous, which is the second genotype, and that means you have one dominant allele, the uppercase letter, and one lowercase allele, the recessive letter. And because the dominant rules over the recessive trait, or the recessive allele, heterozygous individuals will always show the dominant trait, in this case purple. And then last but not least, we have homozygous recessive, and that is two lowercase p's, or two recessive alleles. And homozygous recessive individuals with the homozygous recessive genotype will always be white. So, in case you might have noticed by now, that it's possible to have there are two genotypes where you have the same phenotype. So you can be purple, the dominant trait, and be big P, which the genotype is homozygous dominant. So just to sort of recap, purple is the phenotype, homozygous dominant is the phenotype because it has two uppercase P alleles, and if you have that, you're purple. But you can also be heterozygous, have an uppercase P and a lowercase p, and be purple. And you can't tell by looking at the flower. You can't be like, oh, that's a heterozygous purple flower, and this is a homozygous dominant purple flower. You can't tell by looking at them. Just by looking at them, their phenotype is purple. Okay, so the question is, how can we, if we have a purple flower, how can we determine whether it's homozygous dominant or heterozygous dominant? Okay, and the answer is a test cross. Okay, in a test cross, okay, so you want to know if there's a purple flower that has the dominant phenotype. Okay, you want to know if it's big P and big P, big P. Okay, or you want to know if it's big P, little p. Okay, and to test it out, you do a test cross. Okay, and a test cross is when you cross it with a homozygous recessive. Okay, and then you look at the offspring. Okay, to determine if the purple flower is homozygous recessive. Homozygous dominant, meaning that it has two uppercase letters, or heterozygous, meaning it has an uppercase allele and a lowercase allele. Okay, so this is sort of showing the cross here. We're going to cross a purple flower, okay, which is either big P, homozygous dominant, or big P, little p, homozygous recessive. Okay, we want to know which one it is. We're not sure, and we're going to cross it with a recessive flower, little p, which is little p, little p. And if you look at the first case here, and these are sort of introduction to Punnett squares. So let's say if the flower was big P, so it was a purple flower that was homozygous dominant, okay, and you're crossing it with a white flower that's little p, let's see what we would get. So this is a small Punnett square. We put the alleles on the top. So these alleles are from the white flower, and these alleles are from the purple flower, the homozygous dominant purple flower. And sort of we match up. So the big P goes here, from here, and the little p comes from here, and we put it in the box. And we're going to continue along. You'll sort of see how that happens on each box. The letter sort of carries over. And you'll see that you get all of the plants that you get are heterozygous, because they have one big P and one little p, one big P and little p. But since they're heterozygous, they're all going to be purple. Okay, so if you do a test cross and you cross it with a plant, the purple plant, and you get purple plant with one that is recessive, completely recessive, white in this case, and you get all purple flowers, okay, that means the purple flower was homozygous dominant. Okay, now we're going to do the same thing over here, sort of see the results. If instead of the purple plant being homozygous dominant, it was heterozygous, I'm going to sort of see the difference in the results. So here, there are the alleles up here, are the alleles for the recessive flower, and here are the alleles for the heterozygous purple flower. And we'll sort of show how the Punnett square fills out. This P goes here, this P goes here. Sort of same thing in this box. P goes here, big P goes here, and so forth. And you'll see in this case, we have half of the flowers that are purple, right? Big P rules over little p. And then half of the flowers that are white. So we get sort of a 50-50 ratio here. So if you get 50-50, half purple, half white, that means that this purple flower is heterozygous. So just to recap, test cross is crossed with a homozygous recessive. If you cross a homozygous recessive with a dominant and you get all the dominant trait, okay, that means the organism is homozygous dominant that you're trying to figure out. If you do the same thing and you get half showing the dominant trait and half showing the recessive trait, then the dominant flower that you were crossing, or whatever organism it is heterozygous. This brings us to Mendel's first law of heredity. There are two Mendel's laws. We'll do the first one today, and then whenever we come back to school, I don't know, whenever that's going to be, we'll deal with the second one. And this law is called the law of segregation. Okay, and the law of segregation states that during meiosis, and so one thing just to sort of take a step back is that Mendel didn't know about meiosis, but what his law essentially said is that during gamete formation, or when you're making sperm and egg, the alleles, you have two alleles, and the alleles separate and can go into each gamete. So let's, for example, let's say you're heterozygous. Okay, what that means is when you're forming sperm or egg, depending on if you're male or female, okay, the egg or sperm has a 50% chance of getting the big P allele and a 50% chance of getting the little P allele. They separate from each other. If you remember in meiosis during prophase and prophase 1, which I know we need to go over again when we get back to school because we did that fast yesterday, on these homologous chromosomes, during prophase 1, the homologous chromosomes line up. And if this was a plant, let's say this was the male plant, this would have a big P allele, and this one would have a little p allele. Okay? And what these do, that does not like a little p because it's hard. P's are not a good letter. Let's make it really little p. There we go. That looks like an R, whatever. Okay, so P's are not a good letter for these problems because there's not much difference between the upper and lowercase letter. But anyway, so these are homologous chromosomes. This one has the big P allele, which is for purple, and the little P allele, which is for white. And they're lined up here, so they formed a tetrad, and then they're going to line up in the middle of the plate during metaphase. And then what's going to happen during metaphase is that they're going to separate. Okay? This P is going to go into one cell, and the little p is going to go into another cell. And at the end, okay, half of the sperm and egg cells will have the big P allele, right here, and then half of the other sperm or egg will have the little p allele. So if this was a flower plant and he was mating, okay, he would have a 50% chance of giving the big P allele to his offspring, and a 50% chance of giving the little p to his offspring. When does this happen? So the alleles separate from each other. The mother and father chromosome separate from each other in anaphase 1. So here, once again, let's say we're talking about humans now. We have the gene for eye color. Let's say this is the big B allele here, okay, and then over here, mother gives a little B allele, brown versus blue, okay. In anaphase 1, okay, these are going to separate from each other, okay, and that's going to allow half of the sperm or half of the egg, depending on if it's male or female, to get the big B allele, and the other half of the sperm or egg to get a little B allele. And then there's a 50% chance at fertilization that it will be a big B allele sperm that will fertilize the egg, or a little B allele sperm that will fertilize the egg. But Mendel, interestingly enough, didn't know about genes or anything like that. So imagine if he knew about that, how much stronger his work would have been. So we're now going to take a look at something called monohybrid crosses. In the next video, which I already made. I made this last year, so I didn't have to remake it. That goes through how to do practice problems. On the PowerPoint, you'll see we'll go to a Word document. It says practice problems. You're going to go to that page in a few seconds. But monohybrid crosses look at one trait. Only one trait. So maybe you're going to look at the inheritance of flower color. But only one trait at a time. Mono means one, so that's why they're called monohybrid crosses. Before we do that, this is sort of, to be honest, you could have gone to this at the I know this took about 20 minutes, but you could have gone to this slide, and if you knew this slide, you probably know everything that you need to know. Okay, So going back to sort of humans, just to relate it back to us again. So brown eyes is, whoa, I'm lame. This should be dominant. Very, 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 very lame. This should be dominant. You guys can write it in. Whoa, that's me being silly. So brown eyes is dominant over blue eyes. So there are, in any case, there are 3 possible genotypes. Okay, what should go here? Remember, homozygous dominant. So, and we're going to use B in this case, just for fun. So if it's homozygous dominant, that it means it has 2 uppercase B's. Okay, if it's heterozygous, okay, that means it has an uppercase B and a lowercase b. And then if it's homozygous recessive, that means it gets 2 little b's. Okay, now we need to look at, these are the genotypes, possible genotypes. Okay, now we need to look at link these genotypes to a phenotype. Okay, well remember, if brown is dominant, and this should not be recessive, that's lame. If brown is dominant over blue, then any genotype with a big B is going to show the dominant phenotype. So this one will be brown. It's got both brown alleles. But even though this one has a brown allele and a blue allele, okay, because brown is dominant over blue, these both will have the brown phenotype. Okay, in order to show the recessive phenotype, and that's really important, in order to show the recessive phenotype, in any case, you need to have 2 little letters. Okay, so I have blue eyes, so that means that I have to have 2 little b alleles for my blue eyes. Turns out that both my parents have brown eyes. So in order for that to work, and maybe you can figure this out, they had to be heterozygous in order for them to produce me and my brother, who also has blue eyes. Little b has blue. Alright, so now you're going to go to the next video and do practice problems. If you know how to do the practice problems, then you don't technically have to watch the next video, because attached to the homework after the guided questions are practice problems for you to do, not watch me do. So if you feel like you got the problems, you don't actually need to watch the next video. But if you don't know how to do the problems that are on that homework packet, then you should. Alright, I'll see you guys hopefully sometime this week.